Genetic interaction occurs when two or more mutations in genes interact to produce an unexpected characteristic with one affecting the other. For example, two mutations that would not be lethal by themselves can combine to cause the death of a cell. But the process by which genes and their mutations interact with each other is complex, as some are more likely to interact than others. Now researchers from the University of Toronto have created the world's first map that shows how genes in a cell interact. This paper, published in the journal Science, paves the way for a new way to study how genes contribute to diseases by looking at the effect of more than one gene at a time. Genome sequencing projects are providing an unprecedented view of genetic variation, the authors said in the study. But our ability to predict the characteristics derived from genetic information is made more challenging because of genomes that are not essential to survival. They said a study on yeast 10 years ago found only 1 in 5 genes were essential to its survival. Since then, research has found a fraction of our genes are essential in human cells too. Most genes are buffered to protect the cell from mutations and environmental stresses. This makes it difficult when trying to work out which genes are responsible for certain phenotypes, particularly when interactions occur. Our ability to interpret genetic information to predict inherited phenotypes remains limited, the author said, in large part due to the extensive buffering of genomes, making most individual eukaryotic genes dispensable for life. The researchers used yeast genetics to develop their genetic network. Genes with similar genetic interaction profiles are connected in a global network. In the network, genes exhibiting more similar profiles are closer to each other, whereas genes with less similar profiles are positioned farther apart. We've created a reference guide for how to chart genetic interactions in a cell, said Michael Costanzo, a research associate who worked on the study. The map begins to explain how thousands of genes coordinate with one another to orchestrate cellular life. We can now tell what kind of properties to look for in searching for highly connected genes in human genetic networks with the potential to impact genetic diseases. The finding could lead to a better understanding of genetic disorders. Genetic interactions may also explain a considerable component of the undiscovered genetics associated with human diseases, the authors wrote.